Hello Bandhugan, Namaskar, welcome back to my YouTube channel Sahil Educations. In the last video we have already studied about the structure of sarcomere. Please label the elements just watching the previous video. And in this video we'll be discussing about the structural organization, the structural assembly of this actin microfilament. Okay, so this blue line is the actin microfilament and we'll be talking about this microfilament today. If we see actin microfilament, it has two kind of form. One is the globular actin, which is a singular proteins like structure, and the second form of this actin is like filamentous actin. The single globular proteins comes together with the process of nucleation, and then they prepare a double helical structure. This is called as F actin. So let's see in the board now. See, these are the globular actins. That is G actin, can also be called as G actin. And if we see the F actin, then it will look like the second one is second helix will go like this. So this is a double helical structure or double helical form of actin microfilament. In this microfilament, there are some other kind of proteins which are involved to stabilize the structure of this F actin or microfilament actin. Okay, see, the first one is nebulin, which runs along the length of this microfilament. It acts actually like a skeleton to this structure. This actin filament or microfilament has a skeleton of nebulin protein. The protein is nebulin. Okay, number one. Number two is there are some other kind of proteins like tropomyosin. Remember the name? Tropomyosin. This protein also runs throughout the length of this actin microfilament, but there is certain thing like tropomyosin is present in a dimer form. This tropomyosin comes in a dimer form and covers 7 actin proteins. 7 actin proteins. They covers a 7 actin protein, the tropomyosin dimer. Okay, and then other protein calls, that is troponin. This troponin is attached with this tropomyosin. Troponin is attached with this tropomyosin, and this troponin has 3 subunits. The subunits are T, I and C. That is troponin T, troponin I and troponin C. Troponin T, which helps this troponin to bind with tropomyosin. Troponin I, which acts as inhibitor for the binding of actin with myosin, so that muscle contraction is not going there. Muscle contraction will not be there. Okay, so this I subunit of this troponin molecule or protein is acting as an inhibitor. And then coming to troponin C, this troponin C binds with calcium ion in our muscle fibers, which has been supplied from the sarcolemma tubule, or you can say sarcoplasmic reticulum, and the conformational change will occur in this troponin and the muscle contraction will happen. This will be discussed in the sliding filament theory class. Okay, so this is the basic structure or the structure of actin microfilament. Some other kind of proteins are also present here. We'll discuss this also. The protein is like tropomodulin. The two ends have tropomodulin. which actually fixes or defines the length of this actin microfilament. There are some other proteins in actin microfilament also, like alpha actinin and CAPZ. Okay, these two proteins are present in the Z line, to make the actin microfilament stable. The structure of this actin microfilament is facilitated to be stable by these proteins, alpha actinin and CAPZ. The length is going to be defined by tropomodulin. Troponin has 3 subunits, T, I and C. T is bind with the tropomyosin, I acts as inhibitor, C binds with calcium ion. And then coming to tropomyosin, tropomyosin is present in a dimer form which covers 7 actin molecules or proteins in a single stretch. Then again another dimer of tropomyosin will be like this, covering 7 actins. Okay, and then coming to nebulin, which is a skeleton to this actin microfilament. So that's all for this video. Thank you for watching, and see you in the next video.